to the group.
Molati?
Yes.
I think you can do it because I'm going on the other side.
What is happening?
You can introduce her.
All right. Good morning, everyone. I'm sorry, Lerato, if I watched this because I was not prepared or prepped. Today's journal club is going to be given by Lerato Nephali from University of Johannesburg in the Department of Plant Metabolomics there. I've known Lerato for a few years now through some of the ACGT training events, throughout her master's project, and now while she's doing her PhD. There was some overlap with the project that we did, which looks at biostimulant bacteria and its potential applications in farming. And I think she's going to give a bit of a talk with relation to that work and the potential use of that bacteria of biostimulant bacteria. I'll hand over to Lerato to tell us a bit more about the work that she's been doing.
Sure. Thank you so much, Molati, and yeah, for someone who's unprepared, you did very well. So thank you so much. So as Molati said, I'm Lerato, and I won't repeat the introduction. So today I'll just be taking you through molecular networking to profile the metabolome of biostimulant bacteria. So firstly, I'd just like to apologize for the people who have seen the slides before. But I just thought it would be a good way to show, the application of the molecular networking tools and how we are using them in our lab. And yeah, either way, we learn through repetition. So I hope you guys won't be bored. And this is the outline of the presentation that I will be following. To contextualize the study, I would first likely to introduce you to the biostimulant and metabolomics. Biostimulants are biological formulations that can enhance plant growth and stress resistance by triggering plants' natural processes. And they can be categorized into two, which are microbial biostimulant and non-microbial biostimulants. The non-microbial include examples such as humic acids or humic substances, seaweed extract, and microbial biostimulant include PGPRs, which is the focus of the study, as well as the fungi. The biostimulant market has been exponentially growing as the modern agriculture faces dual challenge of meeting the food security goals and increasing demand for sustainable product and programs. However, the biostimulant products that are currently on the market are claim-based. That means that the primary source of evidence is the assessment of the agronomic trait. Therefore, the exact mechanisms in which this product are used, they are poorly understood. Metabolomics has been proposed as an indispensable omic science that can help elucidate the biochemistry underlying the plant biostimulant interactions. Metabolomics can be simply defined as the comprehensive qualitative and quantitative analysis of all metabolites under specified conditions. And this field has grown exponentially over the years with advancement involving the 4IR equipped analytical platforms, chemometric modeling, as well as the computational tools such as molecular networking. Molecular networking in particular will be focusing on in this presentation has completely revolutionized metabolite identification, which is one of the major bottlenecks in metabolomics. Through metabolomics, we are able to identify metabolites and metabolic pathways that are involved in growth promotion and stress protection. Furthermore, through the quantification of this metabolite, we can then correlate them to agronomic traits thereby helping to elucidate the mechanisms of actions. In order to promote growth or confer protection, this biostimulant bacteria needs to be in communication with the plant. And this figure over here visualizes the complexities that are involved in this biotite interaction. There are metabolites that are produced by the plant into the rhizosphere, which influences the bacterial metabolism, and vice versa. The metabolites that are produced by the bacteria, which are also secreted into the rhizosphere, which influence the plant metabolism. Subsequently, this leads to plant growth and stress protection. However, decoding this chemical intercommunication is still enigmatic. That means that the interactome between the bacteria and the plant is poorly understood. Therefore, this hampers the industries and farmers from confidently and innovatively formulating and fully implementing the use of PGPR-based biostimulant into agronomic practices for sustainable agriculture. Therefore, this study aims to apply metabolomics and molecular networking to profile the intracellular metabolome of the biostimulant bacteria, under the notion that if we can understand the chemical space of the biostimulant bacteria, we can then better understand what metabolites are being secreted and how the communication is being carried out to the plant. And the study was carried out by firstly determining the growth rate of 4 bacillus isolates, which are listed over here, as well as their consortium. And this was followed by culturing of the bacterial isolate as well as the consortium in LB media, and the harvesting was done at 4 different time points, which reflected the lag, the log, and the stationary phase. This was then followed by extraction of metabolites from the bacterial cells, and the extras were then analyzed on LCMS platform. And the acquired data was then processed and used for molecular networking. Molecular networking tools are housed in the NPS environment, and there are many workflows that are found therein which are useful. However, I'll be focusing or highlighting the few that I've listed in here, which are the ones that are used in the study. And this includes feature-based molecular networking, which performs metabolite annotation via library method, and MS2LDA, which performs substructure or fragment annotation, and the MolNet enhancer, which performs class annotation. And I will go a little bit deeper on these tools as I go through the results. And this figure here on figure 2 is the basic principle of molecular networking. Molecular networking, the molecular networking was developed based on the fact that if metabolites are from the same class, they will produce a similar fragmentation. Okay, if we look at this, I'll take you through this process. If you have the sample and you have extracted metabolites from whichever can be a plant or bacteria, and you analyze that sample on an MS-based platform, it then yields many spectra, which are then organized using MS cluster, and the similar or same spectra is then decomposed or collapsed into one node. As you can see over here, each spectra is now corresponding to one node. And this is being followed by spectral alignment, which compares the spectras, and it compares them based on their spectral similarity, which is usually measured using cosine scores. So how cosine scores work is that the higher the cosine score, the higher the similarity between the spectras, and the lower the cosine score, and the lower the similarity between the spectras. And this ends up yielding a cluster of nodes that are connected to one another. As you can see, these 3 nodes are connected, which means they have a similar spectra, and that means they are structurally similar. As you can see, there's a singleton on the side here, which means this molecule is different structurally from this cluster of metabolites. Furthermore, molecular networking also has a built-in library in GNPS, and this now searches this node, the experimental spectra against the library. And if there is a match, for example, if this one in orange is meshed into the library, if, for example, if we have it meshed to a phenylalanine, then we'll know that the other two nodes are structurally similar to phenylalanine. So in this study, we used the computational tools to decode the chemical space of bacteria, and we use feature-based molecular networking as well as MS2LDA and MolNet enhancer. Once again, feature-based molecular networking organizes and aligns the spectra based on the spectral similarity, and also performs metabolite annotation via library matching. MS2LDA, on the other hand, it reveals the patterns of co-occurring fragment and neutral losses termed MAS2 motifs. And I will make an example here in figure 3B. So identifying the substructures can help to identify or assign a name to a node, a molecular node. And we look at this B, this node MZ279. It has a corresponding spectra over here. So MS2LDA identified the MZ or fragment 132, and it identified it as a leucine-related motif. Therefore, I know that in the spectra, it represents a molecule that has a component of leucine in it. I then used this information to work out the difference between the parent ion and that fragment, and then corresponded to phenylalanine. And eventually I was able to annotate this node as leucine phenylalanine. Figure 4, on the other hand, is a MolNet enhancer network, which shows the class annotations in the consortium. A MolNet enhancer incorporates and visualizes the results from different platforms such as feature-based molecular networking, MS2LDA, and NEB. It uses the information from this platform to predict the classes that could be present in the sample that you are looking for. For example, in this one in the consortium, we were able to see these different classes, the organic acid, the phenylpropanate, and polypeptides, in the lipid, the benzoites, and the others that are listed here. This kind of information, it then helps and narrows down the search to assign the names to these nodes. For example, if we look at the green clusters, which are classified as lipid and lipid-like molecules, under which lipopeptides were found, and these were only annotated at a class level. However, for the lipopeptides, the nodes were not annotated yet. Further chose to continue to interrogate the lipopeptides, as they are important molecules in the plant-bacteria interactions. So this figure over here shows the sub-networks that were extracted from the large feature-based molecular networks of the consortium, as well as the individual bacillus isolates. And there were 4 lipopeptides that I was able to identify, and this includes the surfactants, fungicins, iturins, as well as lichensins. As you can see over here, that this network shows that the lipopeptide profiles in these different individual strains are different from the consortium. And furthermore, I was able to annotate different variants of the surfactant as well of the lichensins. And I was able to see that this connected node, there is molecular ions as well as their adduct forms. And if we can just look at this in the consortium, that there are metabolites that were identified, which are surfactant, And through this, we are also able to identify phagosins which are structurally related to surfactant. Therefore, other nodes that are listed over here, they can also be identified if you investigate them further. Yeah. And similar results were also found in the B. licaniformis with the lichenicins, where we found the 3 variants of lichenicins. And one thing again to mention is that even though this 2 B. lichenicin 100 and B. lichenicin M017, they are both licheniformis strains. However, the lichenicin profile is different. In B. licheniformis 100, we found 3 of these strains, whereas in B. licheniformis M017, we only found 1 lichenicin. Okay, this kind of information can then be used, and it can assist in describing and characterizing the chemotypes of agriculturally important strains, and eventually it can help select the functionally significant strains for biostimulant or consortium formulation by evaluating those key metabolites. The timing of production and the distribution of metabolites within microbial population can also provide valuable insight into biostimulant designs. To monitor the temporal changes of metabolites inside of the bacterial isolate as well as the consortium, we then use feature-based molecular networking. As you can see in this figure, at each time point, the network topology is different, and this suggests that the metabolism of this bacteria is different at each growth stage. Once again, we then zoomed into the lipopeptides, specifically in this case, surfactants, to measure how their presence at different time points. And we found out that the surfactant, which has a shorter lipid chain, was only present at the stationary phase and is shown here in purple. And surfactant B showed up in the exponential phase, and it was present in the stationary phase as well. And interestingly, the surfactant C, which is shown here in red, was present at all growth phases. Furthermore, we also performed the feature quantification using FeatureBased, and this is shown over here in figure 6b. And we're able to see from this that this surfactant were mostly abundant at the late growth stages, in which is the stationary phase here. Furthermore, we also performed heatmap analysis to do the relative quantification of lipopeptides in bacterial isolates versus the consortium, and there's also we also chose to show the time profile of this lipopeptides. And from this, we are able to see that the co-inoculation of this bacterial isolate encourages the biosynthesis of surfactants, as you can see over here. However, it discourages the synthesis of lichenicins. If you can see, they are mostly abundant in B. licheniformis single strains. This information can be used in several ways. For example, surfactants have been proven to be involved in recruiting other beneficial microorganisms into the plant's ecological niche. Therefore, it would be beneficial to apply a consortium as compared to the single strains, as this would encourage a favorable microbial community, thereby encouraging synergistic collaboration in the plant microbiota. Furthermore, this information can be used by farmers and production companies such as Omnia, as they will know which strain produces what lipopeptides or any other metabolites. And once they know that, they can investigate further which strains are more compatible, which will produce a more effective biostimulant. And this encourages a more targeted design of biostimulants for specific purposes. So in summary, the key findings from this study is that the co-culture of the bacillus strains the ones that we showed, promote the enhanced synthesis of the surfactant and not of lichenices, and the lipopeptides are found in high abundance in the stationary phase. Overall, this study demonstrates that the molecular networking tools offers unique opportunities to map the microbial chemical space, thus providing valuable insight into molecular information of the microbial system. Understanding the metabolism of this biostimulant bacteria can help improve the formulation of the biostimulants. And this work has been accepted into Frontier of Plant Science. And I would like to just credit Prof. Justin, who helped really very much appreciate his contribution into the study. And yeah, so initially when I started in the introduction, I mentioned that the most of the biostimulants that are currently on the market are plant-based. That means they can see that when you apply this product into the plant, it helps the plant grow and it protects the plant from distress. However, they do not know how this product works. The exact mechanism of this formulations are not yet fully elucidated. And so it is with the consortium that I just spoke about in the presentation, which is a Bastim. It is actually a commercial product, and you can see here, it's here, that is being sold by Omnia, and it is currently available. So I worked on this project in my master's, where I was investigating the effect of this product on maize plants under normal condition as well as under drought conditions. I also worked with Khaleleto and Mott together on this project, and together we are able to propose a framework of mechanisms as they are shown in here in this figure. So without going deeper into the details, I'll just highlight some few points so you can get a better understanding of what is going on here. So I applied untargeted metabolomics looking at the leaf metabolic changes, and Mott applied the targeted approach, and she also performed DNA methylation as well as gene expression on the leaves, and Khaleleto applied both untargeted and targeted metabolomics to explore the submetabolic changes that were due to the application of Bastim. Okay, for example, I use this PAL, which is a gene that is responsible for the initiation of phenylpropanoid pathway. Under normal condition, it was increased, and this was reflected in the leaves where we saw a differential changes in the phenylpropanoids, and this was also reflected in the sap where we saw also again differential changes in the phenylpropanoids. And we also went on to further perform pathway analysis, and we found that one of the key impacted pathways was phenylpropanoid biosynthesis, and this is related to enhanced antioxidant defense. And we also confirmed this by measuring some of the non-enzymatic markers and enzymatic markers as well, and it did correspond to what we found on the metabolite level. Okay. So if you are interested in this study, you can just go look at the studies that I published in Frontiers, and Khali published in a Plant reporting on the SAP, and Mott's manuscript is still under revision into scientific reports, where it is almost accepted. And yeah, and these are the references that some of them that I used. And I would like to acknowledge the following people: heartfelt recognition to my supervisor, Dr. Fidele Tugizimana, once again, Prof. Justin, Prof. Ian Dubry, Prof. Lizel Piata, and Khaleleto and Mott, and also to the MSA communications team. Thank you so much for giving me this opportunity to be presenting some of my work, and I really hope that some, if not all of you, learned a thing or two from this presentation. and acknowledging my funding, Omnia, and also NRF. Thank you so much for listening. If anyone has a question, I'd like to address that.
Thank you so much, Lerato. So
Yes.
if there are any questions in the chat, you can either put it in the chat box or you can raise your hand. So the biostimulant that you're assessing, you say it comes from Omnia, so it's exclusively produced at Omnia.
Yes, as far as I know, it is exclusively produced by Omnia.
So what is the next step after your research with this biostimulant?
I'm not sure I understand. What do you mean by that? Can you please rephrase?
Based upon your results, so from your study, your PhD study now, what's the next step with the biostimulant? Are you going more broad or more specific with the research?
Okay, so I'm also looking into how, like, the intercommunication in the rhizosphere. So we're also profiling the rhizosphere, which is part of what the work that I do here. And we also did a field trial, and which are also looking at how this biostim performs on real life conditions. So yeah, that's where I'm going with it.
Okay. That's very interesting research.
Thank you.
There's quite a team that Fidele has put together there. So are there any questions from anyone in the audience? Perhaps you can also put your email in the chat, just in case anyone wants to contact you to follow up on your results. If there's anyone in the audience who wants to have a collaboration or a project idea on this research, you're more than welcome to contact the speaker. So we want to have communications going.
All right, I'll just put it up now. Right.
Okay, there we go. There's the email. And it'll also be on the recording in case anyone else wants to listen afterwards and they can contact you.
Yes, all right, I'll do it.
That is the hope with all these journal clubs, is that there are communications that open up and people come and approach you and talk to you about your research. So let's hope that happens.
Yes, I'll be open to help anyone who does need, or if anyone needs clarification on my project, I'll be happy to address.
Awesome. Okay, so I don't see any questions. I thank you again so much, Lerato, for your presentation.
Yes. Thank you so much, Shane, for the opportunity.
And soon, Dr. Lerato, I hope.
Yes, soon.
So as before, I just want to say thanks to Malati for hosting, and our communications group. I see only Keke is in the audience. Keke, Anza, and Chanel. Those three have done a lot of work communicating all these journal clubs to everyone. Just thank you very much to you three. And I believe the rest of the month we are going to continue a little bit more into plants. So next week we have a PhD student from Prof. Prinsloo at UNISA. He's going to talk about some more plant metabolomics.
Mm-hmm.
So this month is plant metabolomics, bacteria metabolomics. So we're moving away from human stuff that I've been shoving down people's throats. Luckily, Fidele and Harald have provided some students to talk on that, because I don't know anything or any students on plant metabolomics. So it's always interesting to read something new.
Yes.
And I love how you guys use molecular networking to annotate new compounds.
Yeah, it is quite interesting, but yeah, very challenging as well. So
Challenging makes it more fun.
yeah,
If it was easy, then why would we do it?
yeah. I mean, may I just mention that this work was actually funded by Innovate UK. So I forgot to acknowledge them. My apologies.
Okay.
Yes.
So they're interested in your biostimulants.
Yes. It was like a big project where we're looking at different combination of this single strain and looking at how they interact with one another and the metabolites they produce altogether. So yeah, it's a big one.
Well, that's awesome. I hope there's more interest coming your way, more projects. You can build on it.
I was going to badmouth you to them to say you didn't acknowledge.
Yeah, sorry, Malati.
Okay, so I think we'll end our journal club there. So everyone, please take note of the email, and please do email our speaker. And next week, we'll see you same time.
Right, thank you so much.
Thank you,
Thank
Lerato.
you.
Cheers, everyone.
Hey, bye.